Dear students, today we will talk about plasmolysis. Here you can see plant cells, outer cell wall is there and inner plasma membrane is there. And we are keeping these plant cells in hypertonic solution. We have discussed this in our previous lectures that if we keep the cells in hypertonic solution, then what happens to them? This is an animal cell. If we keep plant cell in hypertonic solution, the same thing will happen as it will happen in animal cell. Here you can see the water concentration is more inside the cell as compared to outside the cell. So water will come out of the cell into the beaker. This is the effect of hypertonic solution on animal and plant cell, and it results in the shrinkage of cell. The same thing we will do here also. Here what we'll do, we'll keep these plant cells in hypertonic solution, and as a result the contents of the cell, specifically water, will start coming out of the cells, right? So these cells will lose water by process of osmosis. What is osmosis? Movement of water from its higher concentration to its lower concentration through a semi-permeable membrane. Right? Here we are talking about plasma membrane. When water moves from its higher concentration to its lower concentration through a semi-permeable membrane, we call it osmosis. So here these cells are losing water by the process of osmosis. Then what will happen? If the contents of the cell, water is going out, then the contents of the cell will shrink. Here you can see that the cell membrane and other contents of the cell, they are shrinking, right? So we are observing a shrinkage here, but the shape of the cell wall is same here also and here also. The cell wall is not showing shrinking. Why is cell wall not shrinking? Because cell wall is rigid in nature, so it will not change its shape or size. As we are saying that it is rigid means anything that do not bend, it is not flexible, so it will not shrink also. Only the contents of the cell and cell membrane, it will shrink. So this phenomena of shrinkage of contents of cell away from the cell wall is called plasmolysis. This phenomena is called plasmolysis. Now, if we boil these cells, will the result of the experiment be same if we boil the plant material before the experiment? So before putting this into hypertonic solution, what if we boil the plant cells? Will plant cells still show plasmolysis or will they not? Answer to this lies in the effect of boiling on the plant cell. Whenever we boil living cell, be it plant cell, be it animal cell, if we boil any cell, that cell will be dead. It will not be living anymore. And dead cells, they don't show osmosis. Osmosis is shown only by living cells. So this process of losing water by osmosis will not take place if we boil these cells before the experiment. So these cells, the dead cells, will also not show plasmolysis. So I hope you have understood the process of plasmolysis shown in plant cell. Thank you.